So in this talk, we're going to talk about genetic issues in invasive and introduced species. So introduced species, especially invasive species, are a major threat to biodiversity. The inadvertent movement of species around the planet is increasing with the amount of trade. You can see here, this is a map of all of the cargo ships in the last year and the routes that they've taken. You can see they pretty much crisscross the entire planet quite heavily. And these ships, these cargo ships, can be bringing in species and moving them between places and increasing the probability for invasive species. So what is an invasive species? An invasive species is an organisms that are non-native and cause harm. They generally cause harm through things like predation against native species, herbivory against native species, competitive displacement, where they push out native species, and disease, where they bring in disease. So genetics is relevant to introduce an invasive species through several different ways. The first one is genetics can be used to help identify source populations of where invasive species came from. Genetics can also be important in determining the probability that a species will become invasive. And genetics can be one tool that can be used in the control of invasive species. So introduced species go through several different phases of establishment to become invasive species. The first one is introduction and establishment, where individuals get introduced to a new area and then they establish themselves. And the chance of spread into an invasive species depends on fecundity, dispersal ability, and adaptability. And all three of these things are related to the genetics and what's in the genes and how that influences these things. The next phase is a lag phase. This is where the numbers stay stable due to things like the demographics of the species. Maybe they don't have a lot of births, the lack of adaptation. So maybe they're introduced to a new area, but they're not really adapted to that new area, so they don't take off. Or maybe a lack of genetic diversity. So usually when things are being introduced, you have small population numbers, which means you don't have a lot of genetic diversity, and you also have a lot of inbreeding. So these factors can help maintain these introduced species in a stable number before they become invasive. And these things are also related to genetics. And then the final phase is expansion and spread. This is where the species does end up spreading rapidly and becomes invasive. And this relates to genetics because of bottleneck. So when this population is introduced into a new area, generally you have a few number of founders, and so it goes through a bottleneck. And the size of that bottleneck helps influence how diverse the population genetics are. So if you have a really big bottleneck where you only have a few individuals that get introduced and spread, your population is going to have low genetic diversity. And also the rate of expansion after recovery from a bottleneck can influence the genetic diversity. So if there's a really fast expansion, then it's going to maintain genetic diversity, but if there's a slow expansion, then it's going to reduce the amount of genetic diversity. So now we're going to talk about several different genetic issues related to invasive species and invasion biology. So the first one is that genetics can be used to help identify source populations. In order to do this, it's important that the native range of that invasive species is thoroughly sampled, and that source populations themselves are sufficiently differentiated so that you can actually assign it back to those specific populations. An example of this is the zebra mussel depicted here, and researchers used genetics to identify where the individual mussels were coming from. They sampled them in the Great Lakes. They were introduced into the Great Lakes, and then they were able to map them back to Eurasia. And what we're looking at here, these little pie graphs, the colors represent the different alleles that are matched back to those source populations. So you can see that here there's gray and black in most of these in these different ratios, and they fall along different areas over here in Eurasia, which map to the Black Sea and the Caspian Sea drainages. So they were able to source those mussels as that's where they actually came from. So another genetic issue is reduced genetic diversity with a question mark. So you would expect that if you have an invasive species, you have a few number of founders, and therefore you would have low genetic diversity, and then this would lead to inbreeding depression and reduced adaptability across that population. And generally this is what is found when you sample the genetic diversity of invasive species and then back and sample their source populations. However, the repeated introduction of invasive species can actually work to increase the genetic diversity of those invasive populations and actually increase heterozygosity. So an example of this is the brown anole, which was introduced from Cuba to Florida. And here you can see these pie charts again, which represent the different allele variations of the genetic markers. And you can see when you look at these populations, they have more than one color. And so they actually have many different alleles represented in them. And when you look back to the source populations in Cuba, they are generally made up of one color. So it turns out that the introduced species or the introduced populations have higher genetic diversity than the native populations. And again, this is because there was multiple introductions over time, and so that increased the genetic diversity. Another genetic issue is adaptation. So if you recall, in the phases of establishment, we have a phase of lag time. This is where an invasive species gets introduced, but then it sits in stable while it either learns to adapt to the environment and then spread, or it doesn't learn to adapt to the environment and then it diminishes. An example of a case where a species came in and then during this lag time they adapted to the environment and ultimately spread are the fire ants of Argentina. So what happened with this, you can see here we have some fire ants, and we're looking at colony, and this has more than one queen in this colony. And what happened is that initially the fire ants came in and they had a type of ant that they call mono that had only one queen per each colony. But then over time, as the density increased, the only way that the ants could survive is to increase the number of queens in a colony. And so they adapted this, and then they were able to become more and more dense. And it also meant that these colonies were really resilient to any kind of disturbance. So that's why fire ants have gotten such a strong foothold in the Southern North America. Response of natives. So native species can also adapt to the presence of invasive species, and they can respond in several different ways. They can respond by avoidance or exploitation. So let's talk a little bit about avoidance. Here's an example of avoidance. Let's talk about the cane toad. The cane toad was introduced to Australia in the hopes that it would reduce the population of beetles that were eating sugarcane. And it wasn't especially successful because it started eating lots of other things instead. And then while it was eating other things, it was also reproducing, and a lot of predators that would eat things like frogs and toads tried to eat it, but it was really large and also was toxic because it is a toad and so it has toxics in its skin. And so it was killing a lot of these predators that were trying to eat it. And so one of the predators, or several of the predators, actually tried an avoidance strategy. An example of this is the red-bellied snake, where over time they developed a strategy where the snake itself evolved to have smaller heads, so it was less likely to eat a cane toad. It also had a decreased preference for them and an increased resistance to their toxin. So it responded by evolving to avoid the cane toads. Another example of this is in New England. They have blue mussels, and when the Asian shore crab was introduced to the area, it's a predator of these blue mussels, and in a very short time, only 15 years, they found that these mussels adapted and they increased the thickness of their shell so that they were less susceptible to these Asian shore crabs. So these are both examples of avoidance. Another example is of exploitation. So here we have the soapberry bug, and initially it was using the balloon vine fruit here, and as its items of eating, it was eating the seeds of this balloon vine fruit. And to get into these hard casings on this balloon vine, it has a very large mouth or beak length. You can see here it's around an average of 9+ millimeters. But then an introduced invasive species was introduced to the area, and that was the flat-podded golden raintree fruit. And these pods were much easier to get into. And so over time, the native species actually evolved smaller beak sizes so that they changed their preferences. They were able to exploit the invasive species. So when we think about invasives and genetic issues, another thing that comes up is the control of invasives, and this relates to genetics in several ways. So first of all, when you target invasives, you want to try to get at them early. And if you think about this and what it means for genetics, it's because when these invasives or introduced species are early, before they've really spread, they usually have small population sizes, low genetic diversity, and low adaptability. So if you can actually eliminate them at this time, you stand a reasonable chance of being successful. Another way that one might control invasives is to use biological methods. This would include bringing in or introducing parasites, predators, or pathogens to help control the population. We talked about this before. Here are the rabbits of Australia, and they were introduced, a virus was introduced in order to help control the numbers of the rabbits. And they found that over time, the rabbits developed a resistance to this virus. So that's something that can often happen with biological control. Either these new species that are introduced develop resistance, or maybe they go to different hosts, and so there can be problems with biological control. Immunocontraception is another way to control invasives, and this is where you inject the females with a protein that coats the eggs, the outer shell of the eggs in each female, and it ends up blocking the sperm from actually getting to the egg, so it can't be fertilized. So these are the green things are the proteins that lock into the egg, and then it blocks the sperm. And this has been useful, and they're using it right now to manage populations of Assateague Island horses. These are wild ponies on Assateague. They are trying immunocontraception. As a side note, when they tried this, it turns out that the female mares, because they were relieved of the stresses of reproduction, they ended up living longer, which meant that they had more horses around longer, and it actually reduced the population size at a very slow rate. So they had to work with models where they kind of adjusted the population viability analysis to choose an appropriate number of females to actually do this immunocontraception with, so that they were able to lower and maintain a low number of horses on the island. Another example of a way to control invasives is to use sterilization. I've seen with insects they do this by putting them in UV light. They sterilize males usually, and then they have to release males in a really high ratio. I think it's a 10-to-1 ratio, so that when females actually do find a male in the wild, there's a high likelihood that it will mate with a sterile male and not produce offspring. And that has been quite successful. Another thing that has happened with sterilization is that they have introduced a gene that changes estrogens into androgens, so it means that no daughters can be produced by males, and this is used in carp and invasive carp in Australia. So they introduced this gene into this population, and it meant that all of the offspring ended up being males, and so you actually end up flooding changing that sex ratio, and you're reducing the number of offspring that can breed, and so you're reducing your effective population size and reducing this population over time. So that's another way to control invasives. They've talked about doing chromosomal changes. So we would take, the idea here is you take like half a chromosome and flip it on its head, and then that chromosome would be incompatible with another chromosome, so it would make those offspring incompatible. This hasn't been tried much, I don't think, and I think critics think that over time this type of mutation would be selected against because it's pretty bad. It means that there's no offspring, so this wouldn't be a really viable option. And finally, when we're thinking about invasive control and how it relates to genetics, genetics can help us identify eradication units. So for example, the brown rat on South Georgia Island. So this is a rat that is invasive to this island. There's a lot of them. They're quite a problem. And if you were to think about how you can get rid of the rats from the whole island at once, it would be this monumental task. How in the world do you do it? But when they looked at the genetics of the populations of rats on the island, they found out that these glaciers on the island actually separated the different populations of rats into distinct units that do not have any migration or gene flow between them. And that's really useful because that means that you can actually just choose one set, like one part of the island and eradicate them there, and then not worry about them mixing or going or being filled in or moving elsewhere. And then you can kind of attempt to control these invasives one piece at a time. So that's how genetics can be useful. And I also want to talk a little bit about hybridization and introgression, because sometimes the reason that a species becomes invasive is because it either hybridizes, an introduced species will hybridize with a native species, or two introduced species will hybridize together, and then they will take off and become invasive. So let's talk a little bit about what the difference between hybridization and introgression are. So hybridization is an ecological term. It's when you have two species that are mated together. So here is an example of the lion mated with the tiger, becomes the infamous liger. But introgression is a genetic term, and it means the gene flow of alleles from one species to another and the gradual mixing of those alleles. And so you can see this here represented by this figure where you have each color represents a different species, and you can see over time there's a lot of mixing. And if you can actually quantify introgression and figure out like an index of how much an individual has experienced introgression, and it can even help you try to figure out if they were first or second or third generation of hybrids. So hybridization and introgression can be detected with many different types of markers, such as mitochondrial DNA, microsatellites, AFLPs, SNPs, etc. And they tell you different things because we know that mitochondrial tells you more about lineages and only the maternal side, and the microsatellites and the SNPs are nuclear markers. When we think about hybridization and introgression, a lot of times this is actually human mediated. So for example, either humans introduce exotic species and then they end up hybridizing, or humans alter habitats. Maybe if we think about marshes, where they add ditches and they change the salinity of marshes, that can actually increase hybridization. Or also if we think about genetically engineered organisms, such as crop plants. So sometimes crops will have genes in them that are genetically engineered to make them resistant to Roundup or pesticides. And if some of that moves into other populations of other plants nearby, such as the weeds, that could be have really big implications for that farm and for the ecology as a whole. And that really hasn't been evaluated much in the literature, but it's something to think about and keep an eye on. That brings us to another type of genetic engineering in fish. So there's been several times where growth hormones have been added into fish, and this can be a concern because those fish end up being larger and then outcompete the smaller native wild fish. And while they're good for fishermen, they actually have lower survival and they have lower reproductive success. So they end up putting out a lot of sperm, and so they end up fertilizing things, but then they actually end up surviving much worse than the natural smaller fish. This is something that can be a problem, especially when there's low food situations, because at low food levels, these fish really are searching for food, and these populations end up crashing, or even have these populations end up becoming extinct, while the normal fish can survive. So these transgenic fish can be a problem. Now, when we think about alleviating introgression, how do you stop introgression? There are several things that you can think about. So you generally try to remove introduced species, you can eliminate hybrids, and you can focus energy on expanding and protecting pure species. But these things can all be really, really hard and so difficult to manage, and so generally the best course of action is to try to get rid of things before they become a serious problem.